Hello everyone, here is Chapter 1 for Pathophysiology. We're going to start on Slide 3 that looks at a basic example of a eukaryotic cell. Eukaryotic cells are much more complex than eukaryotic or bacteria cells. You can see this nice pretty picture of all of the different organelles inside the eukaryotic cell. As discussed on pages 2 and 3 in the textbook, there are many functions that are all necessary for a eukaryotic cell to maintain homeostasis and to be able to survive and function properly. We've got a few of them listed here: movement, conductivity, metabolic absorption, secretion, excretion, respiration in terms of being able to produce energy, reproduction, and then communication. Here is an example of the plasma membrane, and what it shows is the fluid mosaic model, or membrane fluidity. The purpose of this figure is to show how the outer layer of the membrane, right here, the plasma membrane, is composed of lipids that are not attached to each other in a rigid manner. Rather, the many lipids that compose the lipid bilayer simply form the membrane in a layer of lipids that is open to movement. The proteins and other molecules that are also found in the lipid bilayer, the membrane channel protein, this simple protein, glycoproteins, are able to move around the surface of the cell, but are also securely attached within the membrane, so they can't simply just move in and move out of the plasma membrane. This can be called the fluid mosaic model, as I mentioned already. And if you can imagine these lipids here, if you remember, as a kid, you have that toy where you can, a toy of not nails, but little metal something or others, that you can put your hand in, and you can make an indentation of your hand or of any other molecule or object that you place on those metal pieces. Very similar here. Alright, cell-to-cell adhesions. Cells are generally not found by themselves. Alright, we've had micro last semester, and bacteria live by themselves, or they also live together, but they are their own entity. Individual cells work together to form tissues and then organs, and are held together by different types of cell adhesion methods. Listed here are the three major types of cell adhesion methods. Molecules in a plasma membrane, whether that be proteins, glycolipids, carbohydrates, are often a means for cells to adhere to each other. There are also specialized cell junctions that are areas between adjacent cells that are often open for movement of molecules between cells without having to move into, or without the molecules having to move into different environments or into the extracellular environment. And then there's the extracellular matrix, which we'll talk about on the next slide. So, the extracellular matrix is a meshwork of material that surrounds cells and can act as an adhesion method between cells. You can kind of think about it kind of as glue, sort of. The materials within the extracellular matrix contain fibrous proteins that are produced by fibroblast cells and are embedded in a watery, gel-like substance composed of different carbohydrates. The matrix is also composed of collagen, elastin, fibronectin, and as I already mentioned, it is thought of as glue, but it's not solid, so you have ability to move and be flexible. And here's an example of that, where you simply have figure 111 with the epithelium cells here, integrins, which are going to secure the individual epithelium to the basement membrane, and it's within this environment that you have the extracellular matrix, and you see a variety of different molecules in there. Alright, cell-to-cell communication. Tissues and organs combine to create an animal or a person. As mentioned, these cells, tissues, organs need to maintain proper function. Alright, the communication, the excretion, the production of different proteins and molecules. And all of these have to be maintained for homeostasis, or to have homeostasis and therefore survival. In order for these cells to process or to be in the process of maintaining homeostasis, they have to be able to communicate. This is figure 114, and it shows 3 examples of primary ways of communication. The way or method of communication would be through membrane-bound receptors, right here, that interact with a signal molecule. This is the signaling cell, produces a molecule. That molecule travels to the target cell, and on that target cell you have the specific receptor. This is distance, or this will allow for distance between the secreting cell and the target cell, and often will, if it is far distance, all right, then you have remote signaling, and you have a carrier protein that will allow for stability and sometimes even direction, so to speak, of getting that signal molecule to the target cell. All right, then there's communication between gap junctions, which I mentioned on the previous slide, but really what this is it creates almost like a tunnel that allows for movement of material from one cell to the other cell without having to move into the environment. And kind of an example of this in bacteria would be conjugation, where DNA is moved from one molecule or from one cell to the next. But in this case, we're not talking about DNA or nucleic material, but rather proteins or hormones, some sort of signaling molecule that will move directly from cell to cell. All right, cellular communication relies on those chemicals. And so here you have 6 examples of modes or ways of chemical communication. All right, in this first square here, there is contact-dependent signaling, where you see this little circle here. That is the signal molecule, but it is bound to a membrane. So you have to have physical interaction between these 2 cells, one with the signal, one with the receptor. The cell with the signal must travel to and make contact with that target cell. Then you have paracrine signaling. You have 2 cells that are involved, and generally they are going to be in close proximity. So the signal molecule is secreted by the secreting cell, and then you have the different target cells. Autocrine signaling is auto, right? It's self-signaling, so the secreted cell is the target cell as well. And then by definition, moving on to this next box here, a hormone must enter the blood to move to the target cell. So you have this secreting cell here. The molecule gets into the blood, moves to the target cell. Now we start to combine some of the different types of signaling. Here you have a neurohormone secretion, where the secreting cell is a neuron. The signal moves into the blood and moves to the target cell. Then here you take out the blood, and you simply have the nerve cell secreting the signal directly onto a target cell. All right, now, figure 14 and 15 are different from each other, but there is some overlap. So in the first scenario in figure 14, you have membrane-bound receptor here, which would be shown in the neurotransmitter part here in this figure. But you can also have a membrane-bound receptor in the paracrine. Right here it just shows the signal touching the surface of the cell, but you can also have a receptor there as well. All right, metabolism, we all know about metabolism. And metabolism is simply the chemical task of maintaining cellular functions. All right. It's going to involve either consumption or generation of energy. Anabolic reactions are going to use energy, and those are going to be used to build macromolecules from smaller molecules. Catabolic reactions release energy, and these reactions break down macro or larger molecules into smaller pieces. As energy in the form of ATP, it's generated by breaking the bonds, and breaking larger molecules down results in the production of energy. Alright, so ATP is where you're going to have your energy stored. That's created from chemical energy that is stored within the bonds of your phosphates. Right? It's used in the synthesis of organic molecules, used in muscle contraction, active transport, stores and transfers energy. The molecule ATP does. Alright, this is figure 117. Everyone should be familiar with cellular respiration, process in which cells produce energy. Alright, I spent a fair amount of time on this in micro, and you're really probably from at this point down in terms of glycolysis, producing pyruvate, moving into the transition step to produce acetyl-CoA, citric acid cycle, and then moving to the electron transport chain. Alright, this process is very similar in the eukaryotic cell, except you're starting up here in terms of food. So in terms of humans, we're going to ingest food. It's going to be broken down into proteins, polysaccharides, fats, broken down further, amino acids, simple sugars, and that's where it's going to start feed into these different chemical reactions. Alright, this slide we're really going to just skip over. Membrane transport has many forms. You have passive transport, and these are all just basic definitions. But if we move over to membrane transport for active transport, it's going to require ATP, and what this figure shows are some major activities such as endocytosis, and then endocytosis is going to be large particles that get pulled into the cell, get broken down, and then excreted in the process of exocytosis. Now, you've got the definition of phagocytosis. Phagocytosis is primarily referred to or used when talking about immune cells, but phagocytosis and endocytosis used interchangeably. Penocytosis is going to be very small molecules, or sometimes referred to as drinking by the cell. Alright, here you have specific interactions between these ligands and then the surface receptor. So this is just going to show you the process in which ligands are brought into the cell, how they can be utilized, really just a long process in terms of cell signaling. And that is it for Chapter 1. In my mind, it should be more review for everyone. If you have had AMP, if you've had general biology, really this is a review for you. So listen to this a couple times if you need to, or simply look over the slides.